Good afternoon. My name is Jayakumar Radhakrishnan. On behalf of ICTS, I welcome you all to this distinguished lecture. ICTS arranges these lectures by eminent visitors whenever we have an opportunity so that the general scientific audience gets a chance to listen to developments at the cutting edge. So today we are very fortunate to have Professor Michael Lynch from Arizona. His visit is part of this 2-week program, and there's one more week to go. Yes, so thank you for visiting, and now I will let one of the organizers of the program, Kavita Jain, to formally introduce the speaker and conduct the proceedings.
Okay, so good afternoon. So Christian, Sam, and I are very pleased to have Michael Lynch give this distinguished lecture as a part of this ongoing program in polygenic adaptation. So Mike has made several and substantial contribution to several areas in evolutionary biology, including evolutionary mutation rates, the role of gene duplication, and so on. And he has been instrumental in establishing the field of evolutionary cell biology. He has also written several influential books. Some of them are with Bruce Walsh, who is also here. Mike is currently the director of the Biodesign Center for Mechanisms of Evolution at Arizona State, and he has received several honors, including Thomas Hunt Morgan Medal from the Genetic Society of America and the Lifetime Achievement Award from the SMBE. And today he will tell us about principles of evolutionary overdesign and underperformance.
Okay, thanks very much, Kavita. I think there's only a handful of people here that are outside of the top quantitative genetics group, but I've kind of put the talk together that it would be accessible to physicists and so on. So there'll be some sort of elementary review slides for you folks in PopGen and QuantGen. That title might apply to me, as well as the title of my talk, An Apology to the Sloth Biologist in the audience. Some of you are familiar with this man here on the right. He wrote a lot of books. Not all of them were bestsellers, but one of them was. And hopefully a number of you have read this book. There's some great iconic lines in this book. And along with Wallace, Darwin was really the founder of the concept of natural selection, one of the great conceptual advances in all the sciences. And this has become a big problem for evolutionary biology because so convincing have these guys been that for most biologists and certainly non-biologists, there's nothing more to evolution than natural selection. You just go out and look at differences in phenotypes and try and come up with some story to explain this. And it's sort of enveloped in this one line here: a feeble man can do much by artificial selection. I can see no limit to the amount of change to the beauty or complexity of co-adaptations between all organic beings. So there's this general feeling that if it's beautiful and it's complex, it must have been advanced by natural selection. I'll try and convince you that that's not always the case. I'm going to be thinking big across the whole tree of life today. Before getting into more details, I just want to point out that this has been going on since the foundations of life. So there's this big hole in our understanding of evolutionary biology, and that's the origin of life itself. We're pretty sure that life started out as protocells without genomes that were capable of metabolism. I won't go into the details of how this might have worked because we don't know the details, but eventually there was takeover by RNA and then eventually by DNA in this big monstrous machine called the ribosome. And the plausible argument can be made that the ribosome was nothing more than a parasite of the original protocells. That may be somewhat subject to debate, but here's one I think is no longer subject to much debate. This is the origin of the mitochondrion that occurred at the base of the branch leading to all eukaryotes. And we have data, really good data from phylogenetics and bioenergetics suggesting that the primordial mitochondrion was an energy parasite in cells. And although today we're totally reliant on the mitochondrion for making ATP, we're also feeding that mitochondrion a lot of energy as well to keep the trains running. And then finally, another big event in the history of life, especially in eukaryotes and big multicellular species, is the expansion of non-coding DNA in genomes, intronic DNA, mobile element DNA of all kinds of flavors. None of these can be viewed from the host cell's point of view as adaptations. From the parasite's point of view, I guess you would call them adaptations. But we'll be thinking about host cells as we go through this talk. Now, speaking of complexity and beauty, most of you would agree these are pretty beautiful and pretty complex, and you might spend half your life studying, are these anti-predatory adaptations, ways of finding mates, and so on, and never really did the basic biology, and then did a carbon analysis, found out you just wasted your whole career and you collapse in a heap. These are biology's snowflakes. So most of biology is made of proteins, and most proteins are not made of single monomeric subunits like this, but by small aggregates, dimers, trimers, tetramers, and so on. And the kind of, these are called multimeric proteins, and the kind I'm talking about today are situations in which there's no new function built by the interface. The two subunits carry out the same ancestral function, so they're just agglomerated into into a larger component. And what's interesting about these multimers is you can go in now from the protein database into a wide variety of species and just sum up how frequent are monomers, dimers, trimers, and so on in the proteomes of different bugs. And what you'll find is that you get more or less the same L-shaped distributions in all phylogenetic groups, ranging from eubacteria to the archaea, up into us vertebrates and land plants. They're all basically L-shaped. There's a little bit of a zigzag pattern way out here at the right because odd mers are a little more rare than even mers. But there's no gradient in the complexity of molecules, the number of subunits found in different molecules across major phylogenetic groups. And right away, that's a hint of potential neutral evolution. You'd expect the same pattern going on with the same mechanisms that molecular are driving of establishment of interfaces. And you could say, well, over here, maybe you're comparing apples with oranges because bacterial biologists like to study certain proteins and plant biologists study a different subset. But you can zone down into particular molecules. These are all the glycolytic enzymes that have the same function in glycolysis in all organisms, and go in and look at different species in these different groups. And what you essentially find is for each of these enzymes, you can go into any one of these groups and, okay, in the archaea, there's some archaea using monomers, some using dimers, some using tetramers. There's a gap here for land plants. It's because nobody's studied the structure of hexokinase in land plants. But basically, I think if you studied enough species, you would find all these possibilities in some species in every one of these lineages. And that suggests that over evolutionary time, there must have been movement back and forth between alternative multimeric states. And it gets even more interesting than that, because you can often have species that have the same multimeric structure. This is an enzyme involved in lysine synthesis. And in E. coli and tobacco, in both these cases, it's present as a tetramer. You can see they're using, this is, you might call a back-to-back interface and a head-to-head interface. So something's happened over evolutionary time where they've changed the basic phenotypes of these molecules. And you see this over and over again with these enzymes that we call homomeric enzymes. So each subunit's encoded by the same genetic locus. And about 70% of protein families that have been looked at exhibit this kind of variation. So this kind of variation just moves all over the place. It seems to have nothing really to do with adaptation. Now, when you think about evolution, it's a complex science, a complex set of problems. And most evolutionary biologists focus on things down here, things going on outside the organism, finding mates, trying to figure out how to acquire resources, avoiding bad things like predators and pathogens. But a lot of what dictates biology and the behavior of organisms goes on inside cells. So there's this big problem that we all talk about, historical contingency, but it all starts at the cellular level. We're all stuck with ribosomes that life started out with some 4 billion years ago. We're all stuck with ATP synthase for making energy and so on. These big molecular machines that seem pretty clunky, but this is what we started out with. There's no way back any longer. We aren't allowed to break the laws of biophysics. Molecular stochasticity turns out to be a really big deal because many proteins and certainly messenger RNAs are present in just a handful of copies per cell. So individual cells will vary in their proteomes and in their transcriptomes, and that causes environmental variation among cells, phenotypic variation, well, genetic variation. But ultimately, what selection can and can't do in populations is dictated by what I call the population genetic environment, which has got these 3 dimensions here: random genetic drift that I'll define in a second. There's a noise in the evolutionary process. Mutation generates new variation in each generation recurrently, and then recombination rearranges variation within and among chromosomes. And why this becomes important in trying to think about how evolution can and can't occur on different phylogenetic lineages is because all 3 of these dimensions of the population genetic environment vary by about 3 to 4 orders of magnitude across the tree of life. So this thing we call the effective population size scales negatively with the size of an organism. Talk about that more in a few minutes. The mutation rate, total number of mutations arising per genome, scales inversely with this population size measure. And then the recombination rate is inversely proportional to the genome size in different species. That occurs because as you grow genomes, you increase the number of nucleotides in a genome. On average, you're not changing the number of chromosomes in a genome, and there's typically just one crossover event per chromosome arm. So if you double the amount of nucleotides in your chromosome, you reduce the amount of recombination by 50%. So about 98% of all the variation in average recombination rates in a species can be calculated very easily just by knowing the genome size and the average number of chromosomes per genome. Random genetic drift. People from population genetics, you can take a slight nap here for a second, but I just want to emphasize what this process is. It's, and for the 2 or 3 physicists that might be in the room, I know that you guys in physics tend to think about the word drift as the directional force in a stochastic process, and in population genetics, we view drift as the random force, the random noise. And the way to think about this is you have a bunch of genes starting in this tiny population here with frequency 0.5. The environment can only hold these 10 individuals, but the individuals can produce lots of gametes. And then 10 of these get to randomly survive to the next generation. The allele frequencies changed a little bit. And then repeat this over and over and over again, and the allele frequency wanders around over time. And eventually we'll lose one flavor or the other. And that's a very tiny population, of course. And, but the general principles apply to populations of any size. It's just that the magnitude of fluctuations declines with increasing population size. So here's starting at 0.5 frequency, here's fluctuations going all the way up to a frequency of 1, population size 25. Here's 250. You can see the fluctuations are smaller. Eventually you'll get either to 1 or 0. And then 2,500, they're still smaller. But eventually you'll get to 0 or 1. New variation will be created by mutation. But this is a general process that applies to all organisms. And to emphasize again that this is just one realization of a stochastic process. So if we started this process in a bunch of replicate populations, some would drift to fixation, some would drift to loss, and so on. It's a random process unless there's a directional force underlying everything pushing you in one direction versus the other. That's the numbers game, but most of the noise in the evolutionary process isn't just due to numbers of individuals in populations. It's due to the fact that our genes reside on chromosomes. And so this is, again, just a small sample size, but these would be different chromosomes. And these little tick marks would be marking nucleotide positions on the chromosomes that are variable in the population. And that chromosome picked up this blue mutation, which was an advantageous mutation. And whoosh, after a number of generations, that segment, that linkage block of a chromosome, goes to fixation in the population, dragging that mutation along with it because it's tightly linked to it, and leaving all these other chromosomal types behind. That's a problem because that tick mark happened to be another beneficial mutation. Not as good as this one, and so we lost that beneficial mutation. And then that one over there turned out to be a deleterious mutation. but the net effect of this chromosome is still greater than any of the other ones, so it still went to fixation, dragging a deleterious mutation to fixation along with it. Now, if there was free recombination and selection, the eyes of selection could have looked at each one of these sites independently, then we would have fixed the, there should be a green ball here, shouldn't there? No, a blue ball, that's correct. Yeah, we would have fixed both of the beneficial mutations and we would have lost the green ball, the deleterious mutation. That would be perfectly efficient natural selection. That's what drives effective population sizes down. All these linkage effects and these periodic sweeps that just cleanse out all the variation from populations. And it's worse than that because a lot of what goes on is not cleansing it out by a positive selective sweep, but a deleterious mutation will arise on some chromosome, and that will just lead to the loss of that chromosome from the population. Now, the central way of thinking about what I'm going to be talking about today is related to this concept of what we call effective neutrality that was first developed by Tomoka Oda many years ago. And the idea is very simple here, that a lot of evolution is just competing intensities of different processes. And Oda's idea here was that if the power of random genetic drift overwhelms the power of selection, drift wins. But if the power of selection is sufficiently strong, doesn't matter how much drift you win, selection will ultimately win. And so again, we're thinking about a mutation rising in a population, one copy, and wondering what's the probability of eventually reaching fixation in the population. And the rule of thumb here arises from this. If we think about these two alternative alleles having relative fitnesses of 1 and 1 plus S, S would be the advantageous mutation, 1 over or twice the effective population size is the power drift. That's a measure of the variance in allele frequency change due to just chance. And it's if that exceeds the strength of selection, selection's rendered ineffective. Okay, so that's how we think about effective neutrality. Population size is sufficiently small, and it's really hard to fix very, very small mutations of very, very small positive effects. And this becomes important because, as I just pointed out, there's this range of about 5 orders of magnitude of the effective population size across the tree of life, and it scales with the size of an organism to about the minus 0.2 power. We don't know why that scaling factor applies, but that's what the data tell us at this point. Why this is important, if you live down here and and this is where we vertebrates mostly live, if your effective population size, let's say, is on the order of 10 to the 4th, then that means natural selection can't take advantage of any mutation with an effect smaller than 10 to the minus 4th. And it can't eliminate any deleterious mutation with an effect smaller than 10 to the minus 4. On the other hand, if you live up here, that's as good as it gets. A large effective population size in any microbe, somewhere up around 1 billion individuals. But what this means is that in bacteria, selection can be extremely efficient. It can see mutations with effects as small as 10 to the minus 9. And so as a consequence, response to selection can be much more fine-grained, fine-tuned if you're way up here than when you're down here. We expect the response to selection to be much more granular, at least on this scale of 10 to the minus 4 to 10 to the minus 9 selection coefficients. And these kinds of ideas have helped us understand quite a few different aspects of biology. One of them has to do with differences in genome architecture and gene structure in different organisms. So in bacteria, typical cartoon version of a chromosome would look something like this. You have very, very little intergenic DNA. This would be all the coding DNA. And then you'd have, in front of a few genes, you'll have small regulatory regions. But these are really compact genomes. So typically 95% or more of the genome is coding DNA. And then you get into us big multicellular organisms, and this is everything squeezed in here. Most of our genome, the human genome, is only about 1.5% coding DNA. The rest is intronic DNA. Our coding DNA is split up into small islands by giant intronic DNA. There's all kinds of mobile elements strewn throughout the genome that I haven't illustrated here, and complex regulatory regions and so on. And as a consequence, there's a lot of variation in genome size among organisms. So bacteria are down here, cyanobacteria and so on, and they hug this line of 1 kilobase per gene. Almost the whole genome is coding DNA, and there's about 1 kilobase per gene. And then as you get into larger and larger genomes, you can see things start to level off more and more. So eventually you get to the point in land, plants, and animals in the largest genomes, the increase in genome size is no longer due to an increase in numbers of genes, it's due to an increase in non-coding DNA. So how did we get here? I think this explains a lot of different things in eukaryotic genome evolution. Someone wrote a book on this a few years ago, and this is just a list of, it provides sort of a unifying explanation for a diversity of different comparative observations that we've had. But I don't want to talk about that so much today as some other cellular features. Well, I will mention one other thing with respect to this. One of the problems of having a gene structure like this is it's dangerous for 2 reasons. One is if you have a more complex gene, there's more ways to break the gene. So if you're going to have introns in here, you need to maintain with absolute fidelity certain sites that are signals for splicing. Otherwise, you'll have a non-productive transcript. Even non-functional DNA is dangerous stuff to have around. Even if it has no function whatsoever, it can still pick up a gain-of-function mutation and turn your gene out in the wrong time or the wrong place in development. But more importantly, our genes cost a lot more energetically. There's a lot more DNA, there's more transcripts, and so on. And so we got interested in trying to figure out just how costly it is to maintain a gene in a genome, and how close are we to a use it or lose it sort of situation. You know, how much does a gene have to do for you so that it's paying for itself in terms of how much it costs to run that gene per unit time? And the way to think about this is, you know, this is biology. So this is how we make things in biology in a crude cartoon way. Oops. So the way to think about it is we start with food, carbon and energy source, and then from that food we extract what we call precursor molecules. And then those precursor molecules we use to build monomeric building blocks. By monomeric building blocks, I mean things like amino acids, nucleotides, and lipids. Those are some of the main ones in cells. And in doing that, we're modifying these precursor molecules to make new structure, and we use ATP to do this. ATP is the universal currency in biology for energy utilization. We can talk about CTP and GTP in a few cases, but these are interconvertible for the kinds of things I want to talk about today. So that's the direct expenditure of making a monomeric building block. But then there's this other thing that we call the opportunity cost, because normally if you eat something like glucose, or half the time anyhow, you eat glucose and you just take it all the way down through glycolysis, through the Krebs cycle, and just squeeze every last drop out of it to make energy. But if you divert a precursor molecule to make a monomeric building block, that's an opportunity lost in terms of ATP that you'll never recover because you turned it into a building block. The sum of those then give us an estimate of the total cost of different building blocks. So this is just a table for the different nucleotides, AG, and so on. And they're all about, roughly speaking, it costs about 50 ATPs to make each nucleotide. It costs about 30 ATPs to make each amino acid. There's a lot more variation among amino acids. Some are very cheap, and I think tryptophan or phenylalanine are about 30 times more expensive than the cheapest one. And then lipids are very expensive to make. So you might want to think about that when you're investing in more internal membranes and so on. Bacteria use peptidoglycans to hold their cell walls together, and that gives you an idea. The main point is we're able to estimate these costs of building blocks that all cells use. And this is a gift to us from biochemists. You just go to stuff that none of us enjoyed looking at as undergrads, but all these pathways are worked out. Not all of them, but most of them are worked out in biochemistry textbooks. And if you weed through enough of them, you can figure out the cost of these building blocks. But what we need to know is if we can figure out the cost of a gene, and I'll show you some data on this in a second, we can think about how much does it cost just for the DNA associated with the gene. Then the transcripts, all the transcripts, how many transcripts is the gene making? Transcripts have short half-life, so you have to actually keep replacing them all the time in the lifespan of a cell. The same thing with proteins. We can calculate all that, but you have to scale it with respect to the total cost of the cell. You know, it depends on your baseline budget. You know, if you're somebody like me or Bruce, you know, we can afford to go out and buy a $20 dinner or something. But if you're a starving graduate student, maybe your budget's restricted to $2 because your total budget is slower. So we figured out how much it costs to make an entire cell, going from birth to reproduction. And you can think about that, the cost of constructing the cell, and then there's maintenance costs, of course, too, that don't go into making biomass. And both of these scale essentially, you know, pretty close to linear with cell volume for single-celled organisms. And so with information like that, you can then use the cost of anything that you can calculate in a cell, divide it by the cost of the cell. That's the fraction of your energy budget associated with running that gene. And you can, in a sense, that's a measure of the selective advantage to just lose that gene if it's not doing anything for you. And it turns out there's other ways to calculate these building blocks. This is pretty amazing to me. There's this old formula by Karash and Scher from physical chemistry 100 years ago, and they were trying to figure out the heat of combustion of a billion, every known organic substance. How much energy can you get if you just burn it? They came up with this simple formula. And if you just know the number of carbon atoms, hydrogen atoms, and oxygen, it's a measure of the relative degree of reduction versus oxidation of what you're starting with. You can really estimate to a high degree of accuracy the heat of combustion. It turns out that that heat of combustion is tightly correlated with the ATP level cost of making things in cells. I don't know exactly why this is, but I think it's telling us something about metabolism in cells and how good it is in terms of building things relative to what it could be. So, cost of a gene in different organisms. So, I'll only show you data for 3 different species because this is all we have, the data that's good enough to make these computations. But here's the data for E. coli. These are histograms for all 4,000 genes in E. coli. And I've got illustrated here the cost of replication. So DNA level, RNA level, and protein level. And there's the total for genes. And I guess it's about 10 to the 5.5 would be, this is a log scale. That would be the average number of ATPs it costs to run a gene in E. coli. The upper axis here is nothing but the bottom numbers here divided by the cost of building an E. coli cell, which is about 27 billion ATPs per lifespan of a cell. You can think about those as, again, as a selective disadvantage of harboring that gene if it's not doing anything for you. And so you better be getting more out of it than that, or there's going to be a selective incentive to eliminate it. And then those vertical bars there, that's the drift barrier there. So E. coli has an effective size of about 10 to the 9th. And so if you're to the left here, selection wouldn't be able to feel you. But if you're to the right, selection can easily feel you. And the cost of essentially every gene in E. coli is far, far to the right. E. coli can feel just one, in addition to just 1 or 2 additional nucleotides in the genome can be felt in terms of natural selection. So this is a nice explanation for genomic streamlining in bacteria. And then we move on to a couple of other species, usual suspects. We've got a lot of molecular cellular data, yeast and Arabidopsis. And 3 things happen here. One is these histograms move to the right. Genes are bigger, they have introns in them, more complex regulatory regions or so on. And the cells are bigger, so there's more transcripts being made. And then you go up to the top. These numbers are getting, to a good extent, quite a bit smaller. And that's because the cell, the denominator relative to this, is getting bigger. So the pain you feel by having excess DNA is less because it's less of your cellular energy budget. And then finally, the drift barrier is moving to the right here. That's roughly the effect in the vicinity of the effective population size of yeast, and that's it for Arabidopsis. And you can see now that things are shifted to the left of the drift barrier. So Arabidopsis can't feel big insertions in the genome. I believe there's a mitochondrial genome sitting on one of the Arabidopsis chromosomes, doing nothing but just sitting there waiting for some gigantic deletion to occur, I guess. But the excess baggage there can't easily be felt. So if that happened, it would just have to drift out of the population. So that's the general principles about the cost of genes and how we can calculate them. And I realize I probably put the slide slightly out of order. This thing we call the drift barrier, it's related to Oda's idea. And the idea is if you're pushing the same trait in populations of different size in a directional manner, as you go to the left here, smaller population sizes, drift is becoming a more powerful force. And so positive selection gets stalled at an earlier level of the trait performance than it does out here. So expect there to be these gradients in the efficiency of different molecular cellular adaptations. You're trying to get up here to the biophysical limit. We're not allowed to break that. I don't know if that anything is all that close to the biophysical limit in actual biology, but you get stalled by natural selection. And so if you were to follow individual populations over time here, you would wander around the drift barrier. You would get these mean phenotype distributions that are moving to the right as you get to larger and larger population sizes. And we're going to think about this in terms of mutation rate evolution in a couple minutes. But the way to think about this is supposing there's selection for reducing the error rate here. You start out with a really bad situation up here. And so selection can see you, and it's moving you down to this mutation rate into this gray area. That's the drift barrier. And you stay there forevermore. And down here, this is a really good situation. But unfortunately, the selective advantage for being there is not sufficiently great, and selection can't hold on to you. And so you gradually drift back up into this gray area. No matter where you start, this is sort of like a loose absorbing boundary, and you'll stay there forevermore. So there's a lot of drift barriers that I think one can mention in biology that some of us might not always think about in the words called drift barrier, but one of them I've already pointed out, the origins of gene and genome complexity and low population size settings. Protein biologists have talked about this thing called marginal stability of protein folding and binding interface strength. So most proteins all it takes is like one or two key amino acid changes, and they just can't fold. So they're not well protected against folding problems. And the same is true of those multimeric enzymes that I showed you. That's presumably a reason why they're able to slosh around from one form to another. Reduced enzyme catalytic capacities. At one time, there was one paper in Nature that claimed, I think it was triosphosphate isomerase, was the perfect enzyme. It had reached the pinnacle of evolution. It couldn't ever evolve to a greater extent. And now we think it's probably 2 to 3 orders of magnitude less good than would be based on the diffusion limit. And many enzymes are really good. I'm not trying to give enzymes a bad name. You all know what enzymes can do, but they could in fact be made a lot better. Evolution of senescence, that's also in principle a drift barrier if you subscribe to Bill Hamilton's hypothesis for the origin of senescence. And that hypothesis was that you get older and older and older, most of your fitness has been expressed. And so eventually you get to the point, especially you don't have grandparental care or something, that doesn't matter what happens to you after you've stopped reproducing. That's relaxed selection. That's a drift barrier as well. And I'm going to tell you about a couple more today. One has to do with the evolution of error rates inside cells, the mutation rate at the DNA level, but also at the transcript level and the translational level. Rates and efficiency of growth in organisms of different effective sizes and organism sizes also exhibits an interesting pattern. And then I'll end my talk with talking about the origins of large molecular complexes in low population size settings. Some of our complexes we use in eukaryotes are enormously complicated in terms of number of subunits. And really a good example, I think, of overdesign, and I think we have reasons for thinking how that would happen. So mutation rate evolution, I know this is for some review again for people in the room, but just to emphasize how we think about mutation rate evolution. imagine you have a population of cells, and there's every cell has got one or two deleterious mutations, the little red balls, and then a mutator strain comes along. So it's got an elevated rate of input of new deleterious mutations, and that's the rate of removal of cells with one excess mutation. And so that's an input-output sort of way of thinking about things. And this sub-lineage of mutator cells will evolve in an equilibrium level of deleterious mutations. And that's just equal to the ratio of the input to the output. We want to know how much pain the mutator is feeling, so we multiply the number of excess mutations by the effect, and the S's cancel out in the numerator and the denominator. It tells us that a total effect on fitness of a mutator strain is nothing more than the increased deleterious genome-wide mutation rate. It's independent of the effects of the individual mutations. That's likely to be a pretty small number, because if your number is already really low, the best you could do is go to a zero error rate, which nobody does. So you get to the point where there's little room for improvement. So we expect the mutation rate to be subject to drift barrier. We've spent a lot of time trying to estimate mutation rates. In a lot of organisms, we do this with what we call mutation accumulation experiments. Start with a bug, they're all identical. We put them through bottlenecks of single individuals, long periods of time. And then at the end of the experiment, we sequence the whole genomes. And each line, this shuts down natural selection because the population size is 1. And at the end of the experiment, we were able to figure out the mutation rate in these lines. We've done this with a lot of different bugs, both bacteria and a wide variety of eukaryotes. And the bottom line here is we get this plot that I showed you before. There's this nice inverse scaling between the total number of deleterious mutations arising per genome as a function of the effective population size. So if you move to the right here, you're going to smaller and smaller organisms with larger and larger effective population sizes. So us animals and land plants live up here in the upper right-hand corner. So there's this nice negative scaling. And a couple points to make about this is the upper bound of the deleterious mutation rate compatible with life is somewhere on the order of 1. If it's more than 1, mutations are coming in faster to offspring genomes than natural selection can typically remove them. And so us vertebrates and some land plants, we're right up against the wall in terms of the limits to the genome-wide deleterious mutation rate. Physicists like to sometimes argue that everything can be explained based on speed versus efficiency arguments. That does not work here because the bugs with the lowest mutation rates are the ones that are replicating their DNA most frequently at higher rates, so that doesn't work. And then finally, this is a pretty good example of complexity. More complex organisms with more complex proteins like DNA polymerases aren't better at doing things necessarily. More complex organisms are up here, and their DNA polymerases are in fact more complex, but the mutation rate is elevated by 2 to 3 orders of magnitude. Now, another interesting thing about the error rate that occurs at the DNA level is this. We have when we replicate DNA, there's 3 levels of things going on. The first thing that goes to the polymerase plows through and tries to make a Watson-Crick pair, a proper Watson-Crick pair as it goes along, but it does this incorrectly about 1 in 100,000 times. But then, you know, we've got something like Microsoft Spell Checker here, a proofreading domain, and that picks you up another improvement about 3 orders of magnitude. That's not great. The proofreader is not great. It's making about a mistake 1 in every 1,000 times. And then finally, we've got this thing called mismatch repair, which will get you another 2 orders of magnitude. And this looks great. This makes you think people like this concept of complexity and robustness. And isn't this great? Things must have started out this way. And then there's this quantum improvement, and then another quantum improvement in DNA replication fidelity. But that seems not to be the case. There's multiple species of bacteria, for example, that are completely missing mismatch repair and have normal mutation rates. So it's not elevated by 100-fold. We've developed mismatch repair knockout lines in E. coli and run long-term experiments, long-term being a year or 2, in the laboratory, and they evolve just normal wild-type mutation rates without any chance of recovery of mismeasure. It's been completely deleted. So there's a lot of fluidity and apparently a lot of means for changing error rates inside cells. So a little model way of thinking about things here, how this might happen. Imagine you start out up here. This is the error rate. And you've been here for a long time. And then a molecular miracle occurs, and you get something like proofreading. And so that gets you down to here. That's the error rate of proofreading. The product of those is now your total error rate. So this is a quantum improvement in in replication fidelity. But then over time, what happens is that gradually both of these subcomponents get worse over time, and the total performance of the system goes right back to where you were to start with. So what you've got here is a more complex system, but in the long run, you had a big boost for a transient period, but in the long run, you're no better off than you were before. This is what Steve Frank suggested in a paper in PLOS One several years ago. He called this the paradox of robustness. But my feeling is that it's not only nothing is gained, something's been lost, because now to maintain the same error rate that you had before, you've got a more complex machine. So you've got an elevated cost of building and maintaining the system. There's also more ways to break that machine. So complexity, again, this is an example of complexity doesn't bring improvement in terms of fitness for the organism. There's a lot of other ways errors can be made in cells. This is just kind of hierarchy. If you just want to make a good protein, these are all the places errors can come in. And we'd like eventually to measure all of these things in a diversity of organisms like we've done for the mutation rate, but I'm pretty sure we won't get to it. But one of these error rates that we have gotten to is the transcription error rate, the base loading fidelity in messenger RNAs. So many of you remember we have this thing called the central dogma, another strange aspect of biology that we're stuck with as a legacy. Our genomes are made of DNA. We have to transcribe these into RNA. RNA has to be translated into proteins. Errors can occur here and here. I just talked about errors in replicating DNA. So we've tried to figure out a way to estimate the error rate of the rate of putting erroneous ribonucleotides into RNA. This took some work, but we eventually developed a technique that involves what we call rolling circle reverse transcription. What you have to do is you isolate fresh mRNAs and you fragment them and circularize them, and then you do reverse transcription around them. You try and run around them 2 or 3 times, and then you've read each molecule 2 or 3 times. You have to do this because the sequencing error rate is really high. You can't just read raw RNA-seq libraries because almost all the errors are sequencing errors. But with this approach, you just look for things that are present in every read of the thing, and those are your true errors, and all these other sporadic things are the garbage that you throw away. And so we've done that now for transcript error rates. I'm just going to summarize the data. That's where our DNA replication error rates were that I showed you before. That's it for multicellular species, unicellular species, and so on. This is where transcript error rates are. These rates are huge, 10 to the minus 5. So every 1 in 100,000 bases in a transcript is incorrect. Transcripts are transient. You don't feel quite as much pain. You don't inherit mutation like you do in the DNA over time. So those are really high and uniformly high in bacteria and eukaryotes. But what's even higher, this is something else we've gotten into recently, is the translation error rate. This is the rate at which ribosomes make mistakes, put the wrong amino acid into a transcript error. Those rates are on the order of 1 in 1,000 to 1 in 100. So really, our cells are just populations of variable molecules. Every ribosome has got to have a mistake somewhere in it, inside the cell. So a lot of errors going on inside cells, and yet we soldier on, so to speak. Now, one of the things biologists become increasingly interested, especially cell biology, are these things we call scaling relationships. I just showed you this one, scaling relationship of mutation rate and effective population size. This is a famous one relating metabolic rate is function of the size of the organism. This is an interesting one. This is the swimming speed efficiency. The amount of cell lengths you can swim per unit time per ATP invested. And that goes down dramatically with an increase in organism size. Bigger things are taking a lot more energy to just move a unit body length. Michael Phelps, I guess, would be somewhere down here. And then this is another one, growth rates, and this is what I'm going to talk about. The scaling of maximum known growth rates for organisms of different sizes. And this is an interesting one because you get different scaling relationships in bacteria versus eukaryotes. So in bacteria, the maximum growth rate scales positively with the size of cell division in bacteria. And in all eukaryotic groups, these are all unicellular groups, it scales negatively with about the minus 0.2 power. Remember that minus 0.2 power I mentioned before with the scaling of effective population size. And so physicists have tried to explain these kinds of relationships before, and there's lots of debate. Is this a surface to volume ratio thing, or are there fractals involved? And there's lots of debate among these different camps, but I'll just point out that the data don't support, usually it's minus 1/3 scaling or minus 1/4 scaling. But these data are consistent with both of those kinds of models. It's more like minus 1/5 scaling. And of course, the bacteria are running in the wrong direction. And another thing I want to point out here, harken back to my comments about the mitochondria, is that these data are incompatible with it. We call the mitochondrion the, what do we call it? The powerhouse of the cell, right? And it is the powerhouse of our eukaryotic cells. But a number of people have argued that this allowed a quantum leap in the energy available to eukaryotes. And you don't see that at all for similar eukaryotic cells versus bacteria. If anything, the eukaryotic maximum cell growth rates are lower than in prokaryotes. So what about these alternative scalings? Well, we think in prokaryotes, the positive scaling can be explained by a purely physical model, which might make sense given how efficient natural selection is in prokaryotes. And these ideas are being worked out by a student in my lab named Bogi Trikovic. And the observation here is in bacteria, there's a surface to volume issue that arises as cells get bigger and bigger, right? The surface area to volume ratio goes down as you get bigger and bigger, right? Just geometry. And so that's the surface area to volume at scale with that function of the volume. And in bacteria, the envelope thickness seems to be independent of cell size. So the bigger you get then, the less you have to invest in your envelope, peptidoglycan and all those lipids. And that's expensive stuff, as I pointed out before. So you really gain by being bigger in an energetic sense. And that's why the rate goes up with cell size. But of course, it goes down in bacteria. So this can't explain, in eukaryotes, so it can't explain the eukaryotic results. But there's this interesting thing in eukaryotes, as the cell size gets larger, of course, the surface area volume ratio is still going down, but the envelope thickness is also getting larger, and that compensates. So that in eukaryotes, no matter who you are, typically 20 to 40% of your energy budget is just going into your envelope. I really like this because I bought a few houses in my life, and every time you ask a realtor, at least in the United States, I don't know what's like in India, but in the United States, you say, well, how much of my budget should I be investing in my house? And they always say 30%. So I call that the realtor's law. But the bugs figured this out a long time ago, apparently. Okay, a couple other things on growth rate limits. So one of the things we can do is, there aren't that many physicists here, but you've all heard about the spherical cow, I guess. I guess that's a biology analogy, but let's do sort of a spherical cow analogy here. What we want to ask is how good could biology get in terms of maximum growth rates relative to the problems we've been handed down since the dawn of life? And so what we're going to say is the cell consists of nothing but a ribosome. And all you required for replication is for a ribosome to make a baby ribosome. So to make this calculation, you need to know how many proteins and the number of amino acids in a ribosome, and you need to know how fast the ribosome reads the transcripts for those proteins. If you know those two numbers, which we do for a number of organisms, you can figure out how long it takes a ribosome to replace itself. You cannot possibly get any better than that. And so that's the upper limit to the growth rate for bacteria. And you can see that some bacteria are essentially there. They've essentially climbed to the top. And there's obviously some other stuff going on in cells besides making ribosomes, but ribosomes do constitute a large fraction of the cell budget. The barrier here for eukaryotes is a bit lower because, as you'll see in a second, ribosomes are quite a bit bigger, and the translation rate is a bit slower. And so we're quite far from the barrier in eukaryotes. And I should say that this is the same graph that I just showed before, but I've added on all kinds of multicellular lineages here. So the pattern that I showed before extends to multicellular lineages. And so one issue here is you'll recall that both the effective population size and now the maximum exponential growth rate scale with the -1/5 power of the size of an organism. The implication here is that maximum growth rates scale, since they both scale the same way with a third parameter, they scale isometrically. So you increase effective population size by a certain magnitude, and we are expected on average to increase the growth rate by that same order of magnitude. So if this model is correct, what we're postulating here is this missing growth rate potential is due to the gradual accumulation of mildly deleterious growth rate enhancing alleles as we move to the right. And it's not just that the growth rate changes, the efficiency of growth changes as organisms get bigger and bigger. So this is a plot of the total number of molecules consumed in the lifespan of an organism as a function of the size of an organism. And that can be multiplied by 2.7 times 10 to the 17th to get the total number of ATPs that it takes to build a cell. So this is the energetic expense of growing. And this is where it is for unicellular eukaryotes. And then we get into nematodes, rotifers, Daphnia. These are other crustaceans, even small fish and krill. And you can see there's a quantum leap in the cost of making biomass as you get bigger and bigger and bigger. So obviously you get bigger and bigger and bigger, it opens up new ecological niches for you, but it's costing you in terms of the amount of energy requirements. So I'll close with a final set of examples on molecular complexity. And this relates to a concept that's been around for a bit. It's called constructive neutral evolution, and it largely has sort of emanated for the Dalhousie school of thought. And this is the cartoon version because unfortunately we're yet to work out the mathematical theory, but it's easier to present the verbal model in any event. We start out with a molecule A and it's doing its thing, and then some other molecule B takes a liking to it and it starts binding to the interface. Now we got an AB interface. And what that does is it makes a permissive environment on this patch that's being covered up for B for the accumulation of previously forbidden mutations, like hydrophobic amino acid residues. And over time, these will build up. And so you eventually get to the point where now this is an obligatory interaction. If you take B off, A can't function any longer. That's the basic idea. And these guys like to think about this as a ratchet towards complexity. I think it's an open question because if this is a step-by-step approach, you could argue that why can't we take a step backwards as well and there'd be some equilibrium distribution. But there may be some things that you just can't track back once you stochastically get up far enough. But that's the model. There's some open questions, like the argument usually is that, well, what about B? Doesn't B have something to do? Well, maybe B's still able to do its job if it's just hugging molecule A, or sometimes argue there's excess capacity of B in the cell. There's extra molecules of B lying around. Maybe. Because again, molecules cost energy, so you have to worry about that. And then there's this additional problem. If you want to be a multimer, you have to make more copies of everything because there's this encounter problem. If you want to make a multimer, the monomeric subunits have to find each other, so you need enough excess copies, and maybe that's the excess capacity. So I'm going to close with a few examples of this. This is viewed as the poster child of constructive neutral evolution, that big, massive thing called the ribosome. I think all of you know what the ribosome does. It's used translating messenger RNAs into proteins. It has a catalytic core. The business end of the ribosome is 3 to 4 ribosomal RNAs at the heart of the ribosome. And surrounding those ribosomal RNAs is a big population of proteins, and they're sort of like an onion skin model, layers and layers of proteins around the ribosome. And what's interesting as you go from bacteria into eukaryotes is the expansion in ribosome size. So the ribosomal RNAs maintain essentially the same core structure as in bacteria. So this is just one part of a ribosomal RNA stem loop region in Escherichia coli. And then we go into yeast, and it still has the same core structure, but it's added all these adventitious stems and loops. And then you get into human, and you've got more and more and more of these. And you see this repeatedly. So the ribosomal RNAs are much more streamlined in bacteria. What's more impressive is there's a tremendous expansion in the number of proteins, a 50% expansion in the number of proteins in going from bacteria to eukaryotes. And what's even more impressive is the machinery needed to produce the ribosomes. Only a few proteins in bacteria are used for ribosome construction. There's up to 200 different proteins involved in ribosome construction in eukaryotes. So a tremendous increase in complexity. This is an example of the small and the large subunits. This is where bacteria, this is the number of nucleotides in there. This is where all us eukaryotes are. There's no evidence at all that there's been an increase in the speed or accuracy of the eukaryotic ribosome relative to the prokaryotic ribosome. And I just showed you some of those data in terms of our estimates of translation error. Here's another one. Some of you have heard about the electron transport chain. This is how we take food, and then we do this crazy thing of pumping hydrogen ions derived from various chemical reactions. They have food outside the mitochondrial membrane to make this protein gradient that comes back through ATP synthase. This is this crazy turbine that we use to make our ATP. And these complexes are composed of a lot of proteins, mostly nuclear encoded. There's about 50 proteins in total in the complexes in bacteria. It's double the number in eukaryotes. Carries out exactly the same set of functions, but it's a much more complicated set of complexes in eukaryotes. There's no evidence of increased speed, efficiency, accuracy, or overall stability of the eukaryotic complexes. So all of you have heard of those, at least those proteins, but here's one protein that maybe only a handful of you would have heard about. This is another riboprotein complex. It's called RNase P. Its only mission in life, and it's present in all cells, its only mission in life is to turn a precursor transfer RNA into its mature form by cleaving a single phosphodiester bond. That's its whole goal in life. It does it in everybody. And in bacteria, this consists of just one RNA and a single protein in the complex. And then you get into eukaryotes, and there it is. The RNA is sort of buried in a complex of like 10 different proteins. And there's no evidence that this complex down here is any more efficient at carrying out its task than this one is here. So these are many, many examples of increased complexity of big molecular machines as we move from prokaryotic world into eukaryotic world. So just to sum up here, what I've tried to demonstrate here today is that there's, especially when you're thinking about evolution across the big tree of life, there's many of examples where we can't explain things based on natural selection. Many things seem to be due to non-adaptive aspects of evolution. This is true for gene and genome architecture and how it changes across the tree of life. It explains reduced accuracy of DNA replication in more complex organisms. Many examples, more complex organisms are not better at doing things than less complex organisms. Reductions in rates of growth and growth and efficiency in more complex organisms, then this adventitious growth of large complexes in eukaryotes. So the final summary here is what occurs in evolution is dictated not just by what natural selection can do, but by what natural selection can't do. Natural selection is in relentless pursuit for perfection, but it's limited by patterns of mutational bias, power of random genetic drift, the size of mutational effects, and so on. It's also limited by historical contingencies. We can't get rid of ribosomes. We can't get rid of ATP synthase. This puts a fundamental constraint on how fast we can build new proteins, how fast we can make energy inside cells. Unless synthetic biologists start coming up with new solutions, this is what we're stuck with, is life on the planet. What's been surprising to me is that we've talked about effectively neutral evolution at the molecular evolution for quite a long time. People still debate this, but I'm surprised at how common it is at the subcellular and the cellular level. And that can maybe simply because there's many more degrees of freedom to make the same kind of molecular adaptation out of a variety of different parts. There's no evidence that natural selection is relentless pursuit of complexity and robustness, and yet we still see these papers, often by written people in different fields, if that's the goal of evolution in the world, is to make more complex and more robust things. Talk to bacteria about that sometime. And then finally, I want to emphasize that despite these deficiencies, as these changes are made in various branches of the tree of life, it repatterns the whole genomic architecture and the cellular structure. And that opens up new ways of evolving by descent with modification that you didn't have before. So for example, introns arose in the basal eukaryote. They weren't there for tissue-specific alternative splicing. There weren't alternative tissues. But in today's multicellular organisms, that's what introns allow us to do. These are some pictures of my labs over the past few years and my students. Those aren't all current in my lab. And I've learned a lot from all these folks over the years, a lot more than they've learned from me. And we've been very fortunate to have funding from these folks down at the bottom. And then, as pointed out before, I'm in this place called the Center for Mechanisms of Evolution. We're in this giant copper building. Every time there's an electrical storm, we all head for the basement in this building. Fortunately, we don't have many there. And these are some of my colleagues, and we'd be very happy to have any of you visit us sometime. We have a mechanism. We might even be able to help make that happen. But we're trying to focus in on, we're pretty unconventional. We're trying to focus in on mechanisms of evolution, as you picked up, but primarily thinking at it from the level of cell biology, molecular biology, and we'll see where that goes. And we do that from a wide variety of perspectives, biochemistry to biophysics and so on. So thank you very much.
One of the examples of rearrangements that can lead to better complexity would be our neurons, the ion channels in the neurons.
Neurons.
They are super conserved. If you take the potassium channel, the sodium channel, they're supremely conserved across the point when which these systems started evolving. But then, if they're also conserved, how is it that we are more cognizant than that of more primitive species? This is only
How
because
is it what?
we are more cognizant. It means our ability, in terms of what it is trying to do is exactly the same. A dog and a human, it's supremely conserved. Mammals, it's supremely conserved. Firing patterns are conserved. It's just that.
It's an interesting set of questions. I guess I'll let you down that I don't know the answers to them, but I am hoping to think about this in the future. I spent a decade or so on genome evolution and cellular evolution. I got to find something to do with my life now. And so I was thinking about getting into issues of multicellularity, but just to pose a question to other people in the audience. I mean, we think we're the brainy ones, right? And I mean, we're the only ones doing stuff like giving talks, but just in terms of basic accuracy in responding to the environment. I don't know exactly how you do the experiment, but are we really better than a typical bacterial signal transduction system? You know, it's not neurons, but it's the sensory system of bacteria. I don't know how we even think about it, but I think it's a good area for thought in the future.
Hello. Yeah, hi. So bunch of places I didn't follow and kind of overlapping. So for species with low effective population size, I get that how drift would be overwhelming natural selection. But those species also are more involved in sexual reproduction. They have heterozygosity and recombination. So how do we know that the accumulated, the deleterious mutations that they would be having is because recombination or heterozygosity is masking natural selection? At the same time, they're more complex organisms, so there's more layers of hierarchy between cellular and the ultimate phenotype that natural selection is seeing. So is that natural selection at those levels as such is more attenuated irrespective of genetic drift? And so for instance, for phylogenetically closely related species or within a species, if they're sexually and asexually reproducing populations, do we see differences in these parameters like growth efficiencies and all?
I think you just asked me some really good questions. There's quite a few embedded in there. We may have to talk after, but the fundamental issue is there are differences in sexual versus asexual species. One way to think about that is, well, I like to think of recombining versus non-recombining species, and bacteria don't have, you know, the kind of sex we have, meiotic sex. But on a nucleotide basis, they have pretty much the same amount of recombination as we do. So in that sense, there's not really much of a distinction. It's a different kind of sex.
What about heterozygosity?
Well, I guess I'm not clear what you're asking about heterozygosity. The mutation rate and the population size sort of relates to heterozygosity. I mean, as a first order of magnitude answer to your question, remember that the mutation rate scales inversely with the effective population size. And so it turns out that the standing level of variation, at least it's what we call silent sites, is proportional to the product of N, E, and U. And so it turns out to be roughly constant. It's not absolutely constant, but there's remarkably little variation in standing level of variation among organisms with really different effective sizes. And I think it's because things that have really big effective sizes have really low mutation rates. We can talk more about some of these. I guess she's got the mic, or I am the mic. Oh, okay. Now there's three, okay. There's another one here.
So you were talking that transcripts usually have a higher degree of mistakes during the incorporation of the nucleotides. So suppose we are looking at some really important transcripts which mRNAs are transcribed only at the start of a developmental process, and they are long-lived and they have a lot of influence for a very long time. And in that case, is there any mechanism to reduce their any misincorporation?
Yeah, you're asking, I think, if there's differences in transcript error rates associated with the importance of the gene. We haven't been able to find any evidence of that. And we've looked. And for example, you can look at the error rates in the actual ribosomal RNAs versus the mRNAs. And we also can't find any evidence that there's a relationship between the transcript error rate and the transcription rate, the number of transcripts. We've looked at it in like 40 species. So it seems to be a pretty good genome-wide measure.
Sorry. I had a question regarding how you calculated the cost of a gene, and I was wondering, because you were showing in plants, and plants photosynthesis is important in mitigating the cost, I guess, and you have a maximum photosynthesis you can tolerate because of redox of stress, and so how do you incorporate this? Or does it matter at all, or it's just? And my second question is, how do you do to evaluate the cost of a gene that's inducible by the environment, like stress-responsive genes, for example?
Yeah. Well, I don't think photosynthesis, the cost is just based on the biosynthetic pathways for making the parts in the gene, right? And those don't change whether you're photosynthesized or not. So I don't think that matters. The second part was, inducible genes. Yeah, the same, everything we did would apply, but you need to account for how many transcripts are made per lifespan of your organism.
Yeah, but that will depend on the environment itself and
that's
how
right.
often you
So
encounter
the cost
it.
could change with environment. I think that's what you're saying, right?
Well, I wonder how you estimate how often the adverse environment is met. And so I guess an inducible stress gene is going to be very cheap as long as the stress is not there, and become very expensive when the stress is there. And
yeah, if it's stress induced, you could argue that you run non-stress, the cost is just at the level of DNA. You don't add in the mRNA or the protein. And then that phase where you have to turn on, then you have to add in those different components.
inducible, then the cost would be higher than what you have in your graph. I was wondering whether you would have stronger selection on those.
It might be less if you weren't turning it on all the time. But on the other hand, if it's super inducible and you're making a lot of transcripts and a lot of protein, then the cost could go up. Yeah, that's an issue here. Is we're just trying to calculate the baseline cost of a gene. There's a lot of work still to be done here, but the idea is if you're an ecologist or something thinking, whoopee-doo, this gene does all this stuff for my organism, it'd be nice to know how much better off would you be or worse off would you be if you just eliminated the gene entirely? Are you getting more out of it? Because it's easy to see something there and say, this is great. But now if we pull it out, are you actually better off or worse off in terms of your whole life history? Not an easy, it's just a thought experiment, of course. But.
Seems like implicit in ascribing the differences in the efficiency of ATP usage to different strengths of drift is the assumption that the extent to which an organism's fitness is constrained by energy availability is the same across the tree of life. In other words, that the same particular increase in ATP translates to the same little s across different organisms or similar little s's. Is there any evidence to suggest that that's the case? Because naively, it would seem like, increasing the ATP demand in me would have less of an effect on my fitness than in a bacteria. In fact, in the modern human environment, it could even be of benefit.
Yeah, in terms of ATP, what we're trying to calculate is, well, a lot of the bugs that we're looking at are bugs, right? They're unicellular species. And the idea there, but you're right, the implicit assumption here is there's certain traits that we're thinking of as having a universal benefit. And maximum growth rate would seem to be one, although you could probably come up with arguments in certain contexts that you just want to hold everything back. But that's the assumption here, is that growth rate is under positive selection. And in particular, efficiency of growth. It's hard to think, at least for me, maybe you have some ideas, but it's hard to imagine why there'd be selection for reduced efficiency of energy utilization.
Thinking it might be selection for reduced efficiency, but merely that the strength of that selection could differ. which could lead to the same effect.
Is that a question? A comment, okay.